Strategies for disease control in stone fruit. Major diseases of stone fruit species include brown rot, or blossom and twig blight, bacterial canker or blast, shot hole disease, also called Coryneum blight, cherry leaf spot, peach leaf curl, plum pox virus disease, and little cherry disease. These diseases will be presented in detail further in the coming slides. Monilia blossom and twig blight and Monilia fruit rot are caused by different fungi from the Monilia family. The disease concerns all stone fruit species, including cherries, plums, apricots, and peaches, as well as pome fruits like apple, pear, and quince. The fungus overwinters on infected tissues, like on fruit mummies, flower remnants, and shoots, with fruit mummies hanging on the tree being the main source of infection the following spring. Fungal spores are transmitted by the wind, rain, and possibly insects. The main infection occurs from the balloon stage onward by ingrowth of the pollen tube of the conidiasporum into the flower organ. Rain, heavy dew, and a cool weather during flowering favor infection. Injured fruit from, for example, feeding damage or cracks caused by rain are particularly susceptible to infestation. Blossom and twig blight attacking the flowers and twigs is characterized by a progressive browning of flower stalks from the blossom leading to wilted flowers with a grayish spore layer and droughty flower remnants hanging from the fruit spike. In case of heavy blossom infection, an infestation of the fruiting wood is possible, also called tip drought. In this case, bouquet shoots die back from May to June. In apricots and sour cherries, branch tips rapidly die without prior symptoms, and dead flowers remain on the shoots. Monilia fruit rot attacking the fruits is characterized by brown, round rots at an early fruit stage. Infested fruits dry up, harden, and remain as fruit mummies on the tree. Huge damage occurs, especially in years with rainy weather at the time of fruit ripening. Wet and warm weather promotes pathogen infection and rot spreading. Monilia laxa and Monilia fructicola symptoms include pustules without concentric arrangement on ripening fruits, while Monilia fructigena symptoms include concentric circles with yellowish to grayish spore pustules. An important preventative measure in the management of Monilia consists of removing the fruit mummies to reduce the inoculum potential and thus infection pressure. Reducing the humidity in the trees by promoting an aerated canopy or covering the orchard from bloom until after the harvest to protect the trees from rain also helps to prevent infections. Under weather protection, relevant infestation pressure only occurs on susceptible varieties and when relative humidity is over 90% for several days. A further measure is to plant less susceptible cultivars. In uncovered orchards, direct measures against Monilia blossom and twig blight with plant protection treatments can be performed from pre-bloom until the end of bloom prior to rain events. No direct measures are available against Monilia fruit rot. Bacterial canker and blast caused by the bacteria Pseudomonas syringae affect many plants, including all main stone fruit species, cherries, plums, apricots, and peaches. It is especially serious on cherries and apricots. The infection by the bacteria happens through penetration into small wounds via tree pruning and frost cracks, and stomata. Wet weather and changes from freeze to thaw periods are particularly conducive to infection. Infections during the summer cause symptoms on leaves but do not penetrate the trunk. Symptoms of bacterial canker and blast include discolored or necrotic spots on leaves and dieback of shoots and cankers on twigs and branches. Formation of canker and gummosis of fruit trees is the most characteristic symptom of the disease. Narrow, brown streaks extend into the healthy tissue above and below the canker. Cankers are first noticed in late winter or early spring. Certain cultural practices, including the planting of healthy plant material from nurseries, the whitening of trunks with slated lime to avoid frost cracks, and proper orchard hygiene, such as cutting out infected shoots or grubbing attacked trees, can help to maintain a low number and reduce severity of infections. Furthermore, pruning in early spring or summer after harvest instead of winter promotes fast healing of wounds and minimizes the risk of infections. Covering the orchard also helps to reduce bacterial canker infections. Since infections also happen in winter, ideally the trees would be covered all year round, but this is not possible in regions with snowfall. Finally, planting less susceptible cultivars also helps to prevent the spread of the disease. To disinfect wounds, 2 applications of copper hydroxide applied during the dormant stage are recommended. Shot hole disease, also known as Coryneum blight, is caused by the fungi Stigmina carpophila, previously called Coryneum beijerinckii or Wilsonomyces carpophilus. Cherries and plums are particularly susceptible, while infections are also possible in peaches and apricots. The fungus overwinters on infected shoots, fruit mummies, and fallen leaves. Infections are possible from bud break to June under humid conditions and temperatures above 10 degrees Celsius, while early infections leave the greatest damage. Strong infection pressure is possible in years or areas with high precipitation in spring and/or frequent fog formation. The symptoms on leaves begin with small reddish or brown spots with light green or yellow rings around them. The spots dry up and fall out, leaving typical shot holes behind. Leaf infections reduce the amount of photosynthesis, weakening the plant and decreasing fruit production. Trees that are repeatedly infested gradually die. The infestation on the fruits begins as small purple spots that develop into gray to brown-black lesions. Proper management focused on promoting a well-aerated tree canopy as well as orchard hygiene, like removing and disposing of any infected leaves, fruits, and twigs when pruning, can reduce the infection pressure. Treatments with copper or clay-based products combined with sulfur from bud break until after bloom are recommended in case of high infestation pressure. The treatment at bud break is the most important. In problem situations and in persistent wet weather, protection must be provided from bud break until after flowering. Cherry leaf spot can be a devastating disease for cherries, whereby sour cherries are more sensitive than sweet cherries. It also can occur in plums. The fungus overwinters as mycelium in leaves. The main infection period is during the second half of May. Warm, humid weather promotes infection, while prolonged drought dramatically reduces the germination capacity of the spores. Cherry leaf spot symptoms are visible from May on and begin as purple to red blurred spots on the upper surface of the leaf, which may merge with one another. Yellow-white spore-bearing structures become visible on the underside of the leaf. In case of heavy infection, leaves start to turn yellow and fall premature off the tree in August. Affected trees produce fewer fruits the following year due to decreased fruit set. Manage this disease by promoting the decomposition of leaves in autumn through superficial hoeing and/or compost application, as the fungus survives the winter in fallen leaves. Covering the orchard or planting less susceptible cultivars are additional preventative measures against cherry leaf spot. Regarding direct measures, treatments against shot hole disease also have an effect against cherry leaf spot. Peach leaf curl is a disease caused by the fungus Taphrina deformans. The fungus overwinters as pseudomycelium on bud scales and shoots. The infection of the buds happens very early during bud swelling, from the end of February to the beginning of March. When temperatures reach above 10 degrees Celsius, infections are possible as early as January. Humid weather promotes the growth and spread of this disease. Symptoms of peach leaf curl disease first appear as reddish areas on developing leaves about 2 weeks after leaves emerge from buds. Affected leaves become blistered and thickened, causing leaves to curl. Later, affected leaves turn yellow or brown and can remain on the tree or may fall off. When trees are severely affected, the disease can strongly reduce fruit production. Symptoms on fruit can occur, making the surface corky and cracked. To reduce the infestation pressure, thin out and remove infested shoots and leaves by mid-May. Avoid planting highly susceptible varieties. Generally, in early treatment from bud swell to bud break during humid weather and temperatures above 10 to 12 degrees Celsius with copper is effective. A second spray 1 to 2 weeks later, before green leaf tips are first visible as flower buds begin to swell, is recommended in cases of persistent humid weather. Sprays with proper orchard hygiene, as removing infested shoots and leaves, help to reduce infestation. The plum pox virus disease, also called sharka disease, is the most destructive viral disease of stone fruit species and mainly infects plum trees, but also apricots, peaches, nectarines, and sweet and sour cherries. The disease is caused by the plum pox virus, short PPV, whereby different strains of the virus infect different stone fruits. The disease transmission occurs mainly by infected plant material and by aphids in orchards as insect vectors, but can also be spread by tree pruning through not disinfected pruning devices. Symptoms of the plum pox virus disease include lighter ring-shaped spots and marks on the leaves. Infected fruits are noticeably worse tasting and less sugary, unsuitable for consumption. Fruit deformation can also appear as ring, line, or pox-like depressions on the fruits, up to shriveling of the fruits. Damaged fruits fall prematurely to the ground. Planting of virus-free certified material, resistant varieties, and rootstocks are preventative measures. Control measures for the plum pox virus include orchard surveys. The only way to manage the disease is to destroy all infected trees. Treatment against aphids, which are vectors, helps to lower the transmission of the virus. The little cherry disease virus is caused by two viruses, the little cherry virus 1 and 2, and it affects sweet and sour cherries. The disease often occurs in latent form, meaning without symptoms, and is spread by plant material. The little cherry virus 2 can also be spread by a pest called the maple borer. For the little cherry virus 1, no vector is currently known. The fruits of trees infected with the little cherry disease virus initially have a more or less normal fruit development, but shortly before fruit ripening, about 1 to 2 weeks, there is a sharp slowing down of the ripening process. The fruits remain small, are less colored, have lower sugar content, and are tasteless. Due to the lower fruit quality, especially the smaller fruit size, the market value of the fruit is reduced, causing economic losses. Other symptoms include a premature reddening of leaves, less vigor of the affected tree, and a smaller tree volume. Little cherry disease virus can be reliably detected in orchards by laboratory testing. The management of the disease consists in planting healthy plant material as well as monitoring the maple borer upon confirmation of infection with the little cherry virus 2. Direct control measures include destroying all infested trees and protecting trees against the maple borer. These are the references for the presentation. Thanks a lot for your attention.